Hello everyone, welcome to our YouTube channel Agriculture by Crackers. In this video, I am going to tell a new concept of faster breeding in fruit crops, and this is a completely new concept of breeding technique and how it work and what is its concept. Let's talk about a breeding system that uses a genetically engineered tree flowering gene that produces generation cycle of 1 year or less. In this breeding technique, we generally use a early flowering gene inserted in a plant, which reduce this early flowering gene reduces the generation cycle of 1 year or less. But in this breeding, there is a twist. The twist is that the final release for commercial use, the plant is not genetically modified. Means, as we have used early flowering gene, that's why this plant should be genetically modified. But the twist is that the final produced plant is not genetically modified. The concept, then next slide, go for then history. In 2010, the faster breeding technique was discovered in Appalachian Fruit Research Station i- in West Virginia, Virginia, which promote early flowering and fruiting and enable the cycle of tree breeding be accomplished faster than the conventional breeding. In conventional breeding, the juvenile phase is very uh, complicated for conventional breeding. Means if we know the juvenile phase of fruit crop is very high. It is vary from 5 to 15 years. That's the main problem in conventional breeding. But in faster breeding, that's the generation cycle or juvenile phase is reduced to 1 year or less than 1 year. Then there is four eminent scientists was associated with faster breeding. First one is Rolf Scroza. He was a or he is a fruit breeder. Then Chinathambi Srinivasan. Then he is a genetic engineering of fruit crop. He is associated with genetic engineering of fruit crop. Then Chris Dardick is he is associated with molecular biology of fruit quality of stone fruit. Then Anne Chalahan, he was also associated with molecular biology of fruit quality of stone fruit. Then here this is the photo of Rolf Plaza. Here the scientist used 196 transgenic plum plant, but out of 196, 50 of them which show early flowering or fruiting in within 1 year. Then and this is the photo so that showing early flowering of plum or also early fruiting of plum in greenhouse condition. Then this is the some objective of fast track breeding. High yielding variety, fast purpose is high yielding variety which are resistant to pests and diseases, have shorter maturity period, lack toxic substances, mature at the same time, can withstand environmental stress like a drought, flood, high temperature. Nowadays the temperature is too high. That the main concern in fruit cultivation that reduce the production and productivity also in fruit crop. Then have improved quality. Then what is the methodology or approaches of this breeding? First one is gene transformation. As I have already told you in the definition. We have used a uh, early continual gene, flowering gene. First, this round like structure, this is the agrobacterium, and then we inserted the early continual gene. Then we transfer to this agrobacterium to a plant or inserted. Then this plant regeneration, that T indicate that it carry the ECF gene, means the plant is genetically modified. After that, this plant is taken for a parent plant for next generation. Then here this transgenic plant taken as a parent plant, then cross it with high quality line, then we get high quality ECF tree as parent, high quality ECF tree. Then this high quality ECF tree taken as a parent plant for next generation. Then this high quality ECF tree cross with high quality line, then we have get high quality ECF tree. Likewise we go for backcross 3, backcross 4, likewise. Then after the crosses, finally scientists get four type of tree. Out of four type, they got a plant which contain high quality type with no ECF gene. Mark my word, high quality type no ECF gene. Only the high quality non ECF gene types are selected. That's the main concept in this breeding. And as we have used the early continual gene, which reduce the generation cycle of the fruit plant within a year or less. That's why the generation cycle is less. And then what is the outcome of this technique? Let's discuss here. The outcome of this technique is, instead of waiting 3 to 4 years of each generation, in conventional breeding, a generation can be made in every year. That means we can complete a breeding cycle or generation cycle within a 1 year by using this fast track breeding technique. But in conventional breeding, it will take 3 to 7 years to complete a generation cycle. Instead of waiting 15 to 20 years for each new variety, a new variety can be made in 5 years, within a 5 year. But in conventional breeding, it will take 15 to 20 years. Then, for example, just consider a generation time of a plum plant is 4 years, but we need four generation. That would normally require 16 years. You can calculate 16 years, but in using faster breeding, four generation can complete within 4 years. And in the end, selected faster breed tree are not genetically engineered. This is the main concept of the fast track breeding. Then let's see some flowering gene which induce early flowering in fruit crop. Here are some example. This is the leafy. This is the gene which is extracted from Arabidopsis thaliana plant and inserted in the citrus that induce early flowering in citrus plant. Then, but same gene when we inserted this gene in Malus domestica plant, but it is not showing any early flowering. This is the disadvantage of this breeding. Some plants show earliness, but some plant does not show any early flowering. This is the main disadvantages in this uh, breeding technique. So there is another gene, Apatella 1 gene, which is also extracted from Arabidopsis thaliana plant. Then it is inserted in the citrus. Then it induces early flowering in citrus plant. But same gene, when you insert it into Malus domestica, does not show any early flowering. This is the some advantage, sorry, disadvantage in this breeding. Then let's discuss some benefit or advantages of fast track breeding system. This is the first one is greater speed of variety development. As I have told, uh, in conventional breeding, it is like take 15 to 20 years to develop a variety, but in fast track breeding, it will require only 4 to 5 years to develop a uh, variety. Then it is less costly method. Then the final produced plant is no non-transgenic plant. These are three advantages or benefit of this fast track breeding. Then discuss about some disadvantages. In most woody fruit species, transformation and regeneration of early one is not available. As I have told in that table, some plant species does not support that gene. That's why the regeneration of the or regeneration of plant is not occur, or that plant does not support that early gene. That's why the early flowering is not occur. And second point of disadvantages, and overexpressive of or overexpressive of the early flowering gene that may create or that may form um, malformed fruit, and also the seed set in that fruit is very low. This is that was the second disadvantages of the hashtag breeding. And third one is in India, like country, regulation does not permit the fast track breeding because in India only uh, cotton plant which is genetically modified, you that genetically modified plant, the government give the access to cultivate. But as we here we have used that genetically modified gene. That's why India may not permit the fast trial breeding. And here, that's all. Thank you. Stay safe, stay healthy.